Necrotizing soft tissue infection is a serious infection caused by several kinds of bacteria. The most feared one is flesh-eating bacteria, so-called Strep A.
Streptococcus pyogenes, or Strep A, is a very virulent bacteria that can cause a lot of different diseases in the human body. And it can also kill the blood vessels, so the flesh dies, and this is when it becomes a flesh-eating bacteria.
It can develop from minor trauma with or without wound, and from minor surgery. It have a mortality of about 30%, and often it's young people, so it's a very serious disease. The challenge in NSTI is to get the diagnosis as fast as possible, and the final diagnosis is only found by surgery. I have, in the most challenging cases, had to operate every second hour to remove the dead tissue.
One of the problems with flesh-eating bacteria is that we cannot get the medicine to the place where the bacteria are living, out in the dead tissue. And there we hope that phages can help, because they can have enzymes that can break down barriers, and when they meet the bacteria they can start a chain reaction, because they are growing where the bacteria are growing.
A bacteriophage is simply a virus that specifically infects bacteria, and all bacteria have these natural enemies, and they're really useful as an alternative medicine because it's a new way of killing bacteria. They infect, they inject their DNA, they take over the machinery of that bacteria, and then they kill that bacteria. So we know that when patients get necrotizing fasciitis caused by this bacteria, it's a rapid and very, very aggressive infection. So being able to use bacteriophages to prevent that rapid spread could be revolutionary. So the question really is, why have nobody found bacteriophages that target Group Strep A before? And I think they probably haven't looked in the right way and in the right places. What we really specialize in our lab is trying to understand where the bacteria might be present in the wild or in natural environments, and bringing those bacteriophages into cultivation. So we know that Group Strep A is found associated with skin and the mouth of both humans and other animals. So that's where we're sampling first of all. So over the last decade or so, we've been looking for bacteriophages that target pathogens of bladders, lungs, and guts, and then more recently also in skin. So we've been finding banks of bacteriophages that kill all sorts of different pathogens, and slowly bringing them into isolation so that we can work with them to find a whole set of novel therapeutics.
Although phages have this great therapeutic potential, it can be very hard to find the right phages. For that reason, we have designed a new strategy to support the traditional phage discovery by using genomic information and machine learning to identify and utilize new phage-derived products like endolysins and phage satellites.
Endolysins are enzymes that phages use in order to destroy the cell wall of the bacteria. So we are going to get samples from the hospitals, grow them in the lab, extract their DNA, sequence them, and try to identify where are the endolysins inside these genomes. As soon as we find suitable endolysin candidates, we would like to express them through other hosts and assess their efficacy against pathogenic Strep A.
Many pathogenic bacteria contain both phages and phage satellites. Phage satellites hijack the vital particles of the phages, and that's how they deliver the DNA. So using the same mechanisms, we want to deliver genes that will allow us to eliminate the pathogen or even reduce the virulence. Having the sequences of the phage satellites, we can engineer and create other type of phage satellites which will carry the genes that would allow us to treat Strep A infections.
Our hope for this process is to characterize the flesh-eating bacteria and find some markers so we can very fast find them and very fast find a treatment for them.
I hope phages therapy will make a better treatment for NSTI, and in the longer term I hope we can find a PCR test so we precisely know which kind of phages we could use, and then reduce the operations and probably save more lives.
In this project, it's the first time we work together with 3 different Danish hospitals. I hope this really brings the foundation of phage therapy to Denmark.